IB Bio, Option B, Biotechnology, Bioinformatics Part 9 is the first of two movies that look at B.5 Bioinformatics. Part 9, B.5a, this movie will have its focus on bioinformatics. Part 10 will look at the applications of bioinformatics. The essential idea is bioinformatics is the use of computers to analyze sequence data in biological research. Here is the outline of all the movies available for Option B. Use this outline to find the movie you need for review. This movie is focused here, but I need to comment about the B.5 movies. Both movies will only provide a very broad overview of the understandings in the IB syllabus. Most of the work to be done in this section of Option B will be done with the use of active gene databases. So what is bioinformatics? Bioinformatics is an interdisciplinary field that develops methods and software tools for understanding biological data. Bioinformatics combines computer science, statistics, and engineering to analyze and interpret biological data. Bioinformatics uses computer analysis and simulation in the field of genomics, the identification of genes, nucleotide sequences, and amino acid sequences. Bioinformatics has the aim of understanding the genetic basis of disease, unique adaptations in organisms, desirable properties, especially in agricultural species, or differences between populations, otherwise known as phylogenetics. Spend some time with these last two slides and take some notes. Numerous companies are now involved with bioinformatics. The European Molecular Biolab focuses on nucleotide databases, as does the DNA Databank of Japan. Swissprot focuses on protein sequences. The Protein Databank focuses on three-dimensional structure databases. Array Express focuses on microarray databases. The Kyoto Encyclopedia of Genes and Genomes focuses on modeling metabolic pathways. The National Center for Biotechnology Information, NCBI, in the U.S. is the home of GeneBank, a database with both nucleotide and protein sequence information. Databases can be used as a reservoir of information, a place to which new data can be added, or the databases can be used experimentally as a source of data about which one has hypothesized. The growth in genetic information has been exponential. This graph shows base pairs sequences on the y-axis over time since 1980 on the x-axis. The y-axis is log scale. So while the increase in known base pairs and sequences appears linear in this graph, the growth is exponential. The exponential growth in genetic information since the early 1980s is clearly shown in this graph. The number of known base pairs is in the billions, while the number of known sequences is in the millions. The Human Genome Project, completed in the year 2000, sequenced for the first time the 3 billion base pairs of a human. This graph not only displays the number of known base pairs since 1989 in blue, but it also shows the number of users in red of the NCBI GeneBank database. By the way, the largest contributor to genetic information is the GeneBank run by NCBI. The GeneBank contribution to information can be seen in blue relative to the European Molecular Lab in off-white, or the DNA Databank of Japan in red. Cooperation in the access to information among scientists is critical to the forward momentum of the scientific endeavor, not to mention the success of genetic databases. But many scientists are working for private companies where profit is important, thus the information might be protected. The tension between public access and propriety, including patent rights, looms large as genetic information continues to expand. The GeneBank database is designed to provide and encourage access to the most up-to-date and comprehensive DNA sequence information. Therefore, NCBI places no restrictions on the use or distribution of the GeneBank data. That said, some submitters to the GeneBank data may claim patent, copyright, or other intellectual property rights in all or a portion of the data that they submit. As well, some authors are concerned that the appearance of their data in GeneBank prior to publication will compromise their work. GeneBank acknowledges this issue and will withhold the release of new submissions for a specified period of time. So here are the first two IB syllabus statements that over the last 10 slides I've covered. Databases allow scientists easy access to information. The body of data stored in databases is increasing exponentially. The National Center for Biotechnology Information is part of the United States National Library of Medicine, a branch of the National Institutes of Health. NCBI was founded in 1988. The NCBI houses various databases, including GeneBank, and the databases are available online through the Entree search engine that integrates available information. The director of NCBI is one of the original authors of the BLAST sequence alignment program. So that brings us to this IB syllabus statement. Define BLAST and distinguish BLASTn and BLASTp searches. BLAST stands for Basic Local Alignment Search Tool and is a computer software tool for comparing primary biological sequence information, such as the amino acid sequences of proteins, BLASTp, or the nucleotides of DNA sequences, BLASTn. Here is the URL for the NCBI homepage, and the homepage appears like this. Under Resources, you can see DNA, RNA, and proteins, and on the right, you can see access to BLAST tools here. If you click on DNA, RNA on the left, or BLAST on the right, you will go to the next page. If you're interested in sequences, base sequences, or amino acid sequences, this is your start page. Notice BLASTn and BLASTp options, and if you scroll down, look at all that's available at the NCBI site. Finding similar proteins, designing primers, comparing sequences, finding matches to express genes, search for markers, and taxonomy searches. All of this is available at the NCBI site. If you have a sequence that you would like to query, there are input formats known as FASTA format. For nucleotides, bases, the format is easy, A, T, C, and G. But with 20 amino acids, the FASTA format is more complicated when you're doing BLASTp searches. Here are the single letter codes for amino acids. And here are examples of sequences of amino acids for a protein, either as capital letters or lowercase. So let's run a BLASTn using a sequence of bases your lab has recently discovered in an unidentified organism. Click here. You enter your unknown base sequence here, and a couple of things to notice. It's a BLASTn, that's what we're conducting. Other options are, I've chosen nucleotide collection, I've titled my search, and I scroll down and I hit BLAST. The search takes a minute or so, so you must be patient as the database works to find a match for your sequence. Then a page like this appears. This is the top of the page. Sometimes there is information that tells you what you want to know right here at the top of the page. But right now, there's not much here, so we have to scroll down. After scrolling down and reading carefully, it appears that your sequence has a match to the gene for human immunodeficiency virus, reverse transcriptase. You can see that there is some indication that your sequence is a partial sequence for the gene for reverse transcriptase. We've just conducted a BLASTn search. Let's modify our search to conduct a protein BLAST, a BLASTp, with the next syllabus statement extending our progress. Databases can be searched to compare newly identified sequences with sequences of known function in other organisms. Let's run a BLASTp using a partial polypeptide sequence of amino acids that your lab has recently extracted from an unidentified organism. This is the start page. Click here. Like the BLASTn, the query sequence is entered here for a BLASTp, But remember the FASTA format. Your amino acid sequence must follow the input data according to the FASTA format code. Then click BLAST at the bottom of the page. Your sequence matches ribulose bisphosphate in Pisum sativum, among other species. Ribulose bisphosphate is Rubisco, the enzyme that fixes carbon dioxide in the light independent reactions of photosynthesis. It would appear that your unknown protein has the function of carbon fixation in plant species. If I click on the first match, information about the sequence that matched your sequence appears. Notice that we're dealing with a partial sequence here, and the FASTA format for the 139 amino acids is given here at the bottom of the page. Again, this is a sequence that matched yours. As well, there's other information about the original author, and that this particular gene for ribulose bisphosphate was cloned into plasmids that contained an sp6 promoter. Further digging on this same partial sequence of Rubisco, and I find 3 references to sequences that are under patent. Biotechnology raises the ethical question of whether natural DNA sequences can be owned or patented. And then there is the tension between what's publicly shared information and what's not. So that brings us to this statement. BLAST searches can identify similar sequences in different organisms. From the BLASTp search that led you to Rubisco, if you scrolled well down the page of matches to your sequence, you would find matches to Rubisco in many different organisms. Pisum sativum, Lotus japonicus, Medicago truncatula, and Trifolium subterraneum. Shawinella mangrovi, Klebsiella pneumoniae, Ricinus communis, Eucalyptus grandis, Phaseolus vulgaris, Juglans regia. BLAST searches can identify similar sequences in different organisms. Leaving behind BLAST searches, let's move to the last IB syllabus statements of this movie. EST is an expressed sequence tag which can be used to identify potential genes. Genes can be discovered by EST data mining. And to ground our understanding, let's define EST. An expressed sequence tag, an EST, is a small part of an active gene made from a copy DNA which can be used to find the rest of the gene by matching base pairs with part of the gene. The EST can be radioactively labeled in order to locate it in a larger segment of DNA. An expressed sequence tag, the EST, is a small part of an expressed gene used to find the rest of the gene by matching base pairs. The EST can be radioactively labeled in order to locate it in a larger segment of the DNA, and genes can be discovered by EST data mining. ESTs may be used to identify genes in a gene discovery search. There are more than 75 million ESTs now available in public databases, such as GeneBank. The NCBI website can be searched for ESTs. You can see that NCBI defines ESTs as short single-read transcript sequences, and that these sequences provide a resource to evaluate gene expression and find potential variation among genes. Remember that genes can be discovered by EST data mining. ESTs are instrumental in gene discovery and in gene sequence determination. There are now more than 75 million ESTs available in public databases. ESTs are approximately 500 to 800 nucleotides long. ESTs consist of DNA that is complementary to mRNA. That's why it's a copy DNA that is used in the EST. ESTs represent portions of expressed genes. They may be represented in databases as either copy DNA or mRNA sequences. Here is an EST page in NCBI for Ziziphus jujuba. Ziziphus jujuba ESTs are on this page. Notice that there are 76 million ESTs in the gene bank database. Notice that the term library is used. ESTs are tools used to find genes. Notice that these ESTs for Ziziphus jujuba are approximately 800 base pairs in length. As I drill down a bit, notice that one of the Ziziphus jujuba ESTs was submitted to GeneBank from a scientist in China. ESTs are copy DNA molecules, short sequences made from mRNA. The mRNA is collected from cells, and these mRNAs represent expressed genes, and then the mRNA is converted to the DNA copy using reverse transcriptase. Once the copy DNA of the gene of interest has been made using reverse transcriptase, you can see that copy here, you sequence the beginning of the copy to make the 5' EST, or sequence the end of the copy to make the 3' EST. Typically, the EST is made from a conserved region of the gene, a region that does not change much among families. DNA sequences, next generation sequences, from an organism Chironomus riparius, also known as the harlequin fly, are exposed to ESTs. In this case, the ESTs are from a full family of insects known as dipterans. The EST probes that hybridize with Chironomus riparius DNA are the ones that are selected. This ACGH experiment stands for Array Comparative Genomic Hybridization. The EST probes that hybridize with Chironomus riparius DNA, they are selected, and the expressed genes in Chironomus riparius can be determined based on the use of EST probes. ESTs can be attached to microarrays, and then genome fragments from another organism, an organism of interest, a cell of interest, tagged with fluorescent molecules, is then washed over the microarray. The genome fragments bind to the EST probe on the array, and the ESTs find the expressed genes within the larger genome. ESTs can be used to identify diseased or cancerous cells based on the specific gene expression within those cells. ESTs contain enough information to permit the design of precise probes for DNA microarrays like this one that can be then used to determine gene expression. In summary, ESTs are used to identify unknown genes and map their position in the genome, discover new genes, identify expressed genes, and make genomic maps. And that brings us to the end of IB Bio, Option B, Biotechnology, Bioinformatics, Part 9, B.5a.